Hi, welcome everyone. I'm very glad to introduce today Eric Dufresne. Eric completed his bachelor's degree in physics from Yale and continued to the University of Chicago for a PhD. He then took a leave of academia, co-founded a tech company, and worked briefly as a management consultant. He then came back to academia and carried out a postdoc in Dave Waits' lab in Harvard. In 2004, he joined Yale, the faculty between physics, mechanical engineering, and cell biology. And in 2016, he moved to ETH Zurich, where he has been ever since, where he is the professor of soft and living materials. His interest lays at the physics of interfaces, and in itself lays at the interface of physics, of material science, and biology, which brings us to today's topic, which is the phase transition of growing droplets in cells and gels.
Thank you very much,
It is yours.
Yeah, thank you very much. It's a great pleasure and honor to present in this series. It's amazing that my first trip to Israel was almost exactly a year ago, and it really seems like a decade ago now. So much has happened in the last year. I had a great time at Weizmann last year, and I'm glad to meet a broader community of Israeli scientists today. So before I start, I need to make sure that I point out the work, the people that did the real work in what you'll hear today. And the most important person to point out is Robert Stile, who is a senior research scientist in my lab, who's been with me for some time, who's been in my main intellectual partner in pretty much everything. And Katie, Tom, Sai, Jin Young, and Alba also contributed a lot of data to this talk. So yeah, so jumping into this is a talk, I think that what you'll see, is pretty broad. It starts with some biological motivations, and then it gets into what I think is some pretty interesting physics and mechanics. And but the original motivation, and to take the really big picture, is asking the question, where do structures come from in biology? And when you talk to a biologist, if you say structure, a lot of times they'll assume that you're referring to the conformation of a protein. And when we try to model protein conformations as physicists, we really use the tools of molecular physics, and the framework for finding protein structures for the most part is the minimization of a 3 energy and using thermodynamics, okay? So that's the physicist view on sort of that classical protein structure problem. On the other extreme of length scales within an organism, it's really interesting to think about how the whole organism develops. And, there's a different framework in developmental biology. And, there when we do modeling, we use a completely different set of principles. We don't refer to energy at all. We think about kinetics, we think about reaction and diffusion. And you could really say that the thing that determines how everything comes together is the exchange of information. So there's a completely different concept. Let me see if we can find my pointer here. Yeah, here's my laser pointer. Yeah, so in this movie on the right-hand side, let's see if I can get it to play. You see a really beautiful oscillatory reaction. Well, you're not seeing the, well, I'll try it again. Here we go. There we go. There's the tail elongating, and as the tail elongates, you can see this oscillatory chemical reaction creating the vertebra. And that's, I think, a very prototypical example of how reaction and diffusion work together at larger length scales to define structures in biology. But there's a whole lot of stuff in between, and that stuff in between is the stuff we love as condensed matter physicists in general and soft matter physicists specifically. And there's more than one answer about where structures come from in biology at those intermediate scales, things that are at the supramolecular scale. But today I want to refer to a very familiar concept to you and show you how it's powerful in biology. The first example that I know of that's really based on data of this idea is this beautiful example by Malcolm Steinberg, originally articulated in the early 1960s. And he called the differential adhesion hypothesis. And he was trying to describe how the cells inside of an embryo sort each other. So, you know, every different type of cell has a different function, and they have to be in specific locations. And he did some experiments where he had two different cell types, and he basically would randomly mix them together. And he found that very deterministically, one cell type would go to the outside and the other cell type would go to the inside. Or there would be one cell type on the right in a little ball and another cell type on the left in a ball. So yeah, that's cell sorting. Another thing that happens at a different length scale inside of a single cell is the formation of a membraneless organelle. So I'm going to show you a movie of an example here. You can see sort of some hazy gray outline of a cell. Right now, that's one component of what's going to become a stress granule. When we stress the cells out, it causes them, as part of their response, to form these regions that are enriched in that particular protein that we've labeled as a bunch of other proteins, as well as a bunch of other proteins and nucleic acids. If you really watch the formation of these objects inside of the cell, it looks very much like a process of nucleation and growth. And after those nuclei have formed, you can see that these objects can coalesce with each other, and they have a lot of behavior that looks very similar to a molecular liquid that we're familiar with. Both of these objects, these two length scales, both involve a common thread of differential adhesion. So on the left, the red cells like to be next to the red cells, but not next to the green cells. And similarly on the right, that fluorescently tagged protein likes to be with itself as well as some other things that are unlabeled, but not with the rest of the cell. That's a differential adhesion part. And then they also have mobility. So on the left, the cells can crawl around and sort of explore and find the partners that they want to be with. And on the right, you know, to first approximation, these molecules are just undergoing Brownian motion and finding their appropriate, their friends that way. Okay. Together, you know, they're both examples of what I would call very generically phase separation. Unfortunately, now in the lingo that sort of spread, cell biologists, when they say phase separation, even in art, there was a huge editorial piece in Science last week where phase separation among the biologists has become synonymous with liquid phase separation near thermodynamic equilibrium. Okay, but phase separation is a much broader phenomena. You know, if you start with Gibbs's definition of a phase, right, and you have two of them that are different next to each other, that's phase separation, right? So here, when I say phase separation, I'm trying to take a broader view in the introduction. Later, I'll move on to something that's actually specifically liquid phase separation near equilibrium. Okay, so this is, you know, one way, alternate way at intermediate length scales biology can organize itself. We're going to dive into this particular method, and you know, I'll remind you of the forces that drive phase separation. I've already referred to differential affinity, and if you're near equilibrium and those affinities are weak, you might get liquid phase separation. But that's not the only thing, as I just alluded to. You know, as we've learned from the community of people working in active matter in recent years, that even if you have objects with just steric interactions but swim at different speeds, shown on the left, macroscopically they can phase separate on the right. And another factor that doesn't really drive phase separation, but can be a really important factor in the way phase separation works, is elasticity. And that's really going to be the focus of my talk. Elasticity in phase separation is actually a pretty old idea. It was first proposed that I'm aware of in a paper by, of course, de Gennes in a very abstract way. And then it was really manifested itself in the whole field of diblock copolymer self-assembly. And then the idea here is that when you have, you know, a polymer with two different blocks covalently bound at one spot, macroscopically, those two blocks might want to be completely phase separated, but because of that microscopic constraint, they're not free to do so. And that elasticity enforces all sorts of really interesting morphologies in micro phase separation. Okay. Now, sort of building off this idea from Dufresne that elasticity can be important in phase separation, somehow, most of us have focused on, we focused on what I would call sort of short range elasticity at the scale of molecules. But what I'm really interested in is how long range elastic interactions, supramolecular, continuum scale elasticity can couple into phase separation. And my lab is working on this in two contexts. Today, I'll only tell you about the context on the right, and that's the elasticity coming from a three-dimensional polymer network. We're also doing a lot of work shown on the left on two-dimensional lipid bilayer systems and phase separation coupled to a membrane. But today we're going to focus on phase separation inside of a polymer network. Now, so let's go back to this movie that I showed you to introduce the idea of a membraneless organelle and stress granules. This process that I described to you of one that looks like condensation and growth. The important thing to remember is that this is not happening in a Newtonian liquid, right? It's happening in a complex and crowded environment. There are many things about the environment of the cell that make it complex. One aspect is the rheology of the cell. The cell has complex rheology, and one of the reasons why cells have complex rheology is because they have embedded polymer networks, okay? And on the right, I'm showing you one example of a cell, a pretty flat cell where we've labeled the microtubules in red. And that process of phase separation that you're seeing on the left is happening inside of this network of microtubules. And if I add to that same image the locations of these little droplets that are forming, you can see they're closely associated. They're right next to this filament network, okay? And if you sort of compare the images on the left and on the right, this big stress granule appears to be located in a big hole inside of the network, right? And so there are all sorts of interesting questions that arise here. First of all, that's an n=1. Is that a statistically significant effect? We'll discuss that in a moment. There's other interesting questions saying, are the stress granules just optimistic and growing in places where there is room? Or really are the stress granules have a non-trivial or interesting mechanical coupling to the polymer network and are interacting with it? So, this is a topic that we're not the only people thinking about this. Cliff Brangwyn at Princeton's thinking about it in the nucleus, and Alexandra Zadowska is also thinking about it in the nucleus. But the basic question they're working on is the same. Okay, and so the way we do it is we look at hundreds of cells, and we trigger the formation of these granules, and at a certain time point, we locate all of the granules inside of the cell, and we sort them according to size. And then we collect an ensemble of these granules that have roughly the same size. And on the left, you can see four just examples of stress granules that have a diameter, of about 1.3 microns. And we have three-dimensional confocal stacks, so we've located these things in 3D. But we can also image what's happening to the filament channels in the cell in the same exact location. And we can ask statistically, how are these structures related to each other? And, we've been, statistical measures are a little bit tricky inside of the cell because it's so heterogeneous, and we've needed to do some work to do meaningful statistics that I'm not going to describe now, but we'll have a paper coming out hopefully on the archive in the next month or so, describing how we get to the data I'm showing you on the right. But the data you're seeing on the right, you can really think in terms of the way it's normalized as a pair correlation function. So on the SG image on the left with n=521, that's showing us what the average intensity of the stress granule channel in the microscope is, of the label, normalized to the intensity in the cell on average. So this is an average over 521 stress granules. Intensity 1 is the average inside of the cell. So you can see inside of the stress granule, that protein is enriched by roughly a factor of 7 compared to the rest of the cell. Now, what's really interesting, and the thing that we want to study, is how this is coupled to the cytoskeleton. And on the right, you can see one example of how it's coupling to the microtubules, right? You can see that there is a strong enhancement of microtubules near the surface of the stress granule. It looks like there's a depletion of stress granules in the middle. It actually has a value of 1, saying that the average intensity of our microtubule marker is the same inside of the stress granule as everywhere else. But there's certainly a big change. What I'm going to do is I'm going to turn this into a G of R. So I'm going to average these images in the angular direction. And you can basically really think of this as a G of R, And there's a peak for each of them. That peak corresponds to the edge of the stress granule. So what we see is a strong increase in the probability of observing a microtubule at the edge of a stress granule. And that increase in the probability gets bigger and bigger as the stress granule increases in size. Okay, so that's the basic statistical observation that we're seeing. Okay, so we're trying to develop some simple models to describe, we're doing some molecular perturbations and all those other things, but I'll tell you about the simple models since this is a physics audience. We thought about a few different things, and we're doing this mostly numerically with very simple models. We think in both cases, we think of the microtubule network as a bunch of straight lines because they have a long persistence length. And then we can just put droplets in randomly inside of the network and put them anywhere that, as long as they don't overlap with a microtubule. And when we do that, you get what you would expect. You get G's of R that show a repulsion, and that repulsion can be pretty long ranged when the stress granule is pretty big. So that, clearly doesn't fit our data at all. Something that works a lot better, but not perfectly, is that if we pick a random location inside of our polymer network and start growing a droplet, and we keep letting the droplet grow until it runs out of room, okay? And in this model, the network is not deformable, but the center of the mass of the droplet can move to eventually it finds a point where it has force balance, or 3 contacts basically, and it's isostatic and it can't move anymore. And if we always have a scenario where we grow until we hit the microtubule, then you can see that we get this nice increase in the microtubule intensity at the surface of the droplet that is consistent with our data. So, however, this very simple model suggests, that there is some sort of I'm not sure if it's there is an interaction, a non-trivial interaction between the droplets and the microtubules, but it doesn't fit the data perfectly. Most importantly, in the simple model I've described, the strength of the correlation drops as the droplet gets bigger and bigger. So bigger droplets accumulate microtubules less than small ones.
Eric?
Yes.
Eric, can I ask a question?
Yeah, sure, Roy.
Hi. One thing about the normalization, in a sense. So what we see on the left G of R, is it just because of to scale all of them? But it's the tail is long enough, and you measure it, so it will all go to 1. So that's maybe the first question.
Yeah.
Because it's surprised to me. I think that the integral of that should somehow converge for all of them, right? And somehow it doesn't look like that. So nothing goes below 1. In any model that I can think of,
yes.
there should be something below the 1. So we'll have so what I'm missing here. So I'm sorry.
Yeah, no, that's a great question, and I think we should discuss it at length, and maybe I'll say a little bit now, and then maybe we could follow up afterwards. So the first thing is, remember that there are the ways that this is different than a normal G of R. It's normalized like a G of R to saying where a value equals to 1 corresponds to the average intensity that you would see in the cell. That's the thing that's most G of R like, and it's a type of cross correlation, because all of these, they're really intensity profiles that are centered in the location of a stress granule. So it's, in a way, it's more like a G of R that's convolved with the density of the object than just the Dirac delta functions. And yeah, this point that you make, that's a very nice one that I'm confused about as well, is, there's this intuition that when G of R goes up in one place, it must go down somewhere else. And let's talk about that some more at the end. Yeah. So anyway, so here we go. Yeah. So another point is, yeah, you shouldn't interpret this like a G of R. It's just normalized like a G of R, right? This, yeah. Anyway, there's so many things I could say about why you should be careful with this. So anyway, since the fact that the correlation drops as the things get bigger, we think this is really a hint, and that we're going to try to quantify this better. But we think this is a hint that as the droplet's growing, it's deforming the network. Okay, and that we're basically, we think what's happening is that the droplet grows, that it's accumulating more and more microtubules. Anyway, okay, so that's where we are with cells, because cells are really hard to do anything carefully. It's easy to be careful with. It's hard to get something that's easy to interpret with a cell. So we're still working hard with cells, but from now, for the rest of the talk, I'm going to shift gears and talk about some polymeric systems where the physics is much more tractable. And I think I see a question from Natalie.
Right.
Yeah.
Yeah, thanks. Just before you move away from the cells, a short question about the dynamics. So if you follow the dynamics, for example, if you can destroy the microtubules and let them reassemble, it should give you plenty of information, right?
Exactly. So there's two types of experiments that we're on the cusp of pursuing. One, right, is exactly what you said, trying to remove the microtubules and or bringing them back. And the other thing is to be able to use these new optogenetic tools like people like Prof have developed, where you can selectively grow a stress granule at a particular location. So yeah, so in the future, everything I'm showing you here is on fixed cells. And now we're in the process of doing experiments with live cells. We have several terabytes of data, and we're just scratching our heads now to figure out how to process it. But those molecular perturbations are key. And I see Sam has a question.
Yeah. Is it clear that when you extract these, that you're extracting only the microtubules and not other parts of the cytoskeletal network like actin that are much less rigid? And the part B of my question is, and that could relate to the second part of your talk, well, I don't know what it'll be, is that the problem of a growing or an object that exerts force on an elastic network and how that mediates interactions between many such objects, is known. I'm sure you know, it is sort of a part of what's called the Eshelby problem in elasticity. And elastically mediated interactions by little spheres that exert force might be relevant, but it could be the microtubules, but in this case, it could also be a more elastic network like the actin.
Exactly. So in the grand scheme of things, there's so many great things to say about that, is we are systematically trying to look, we have already looked at microtubules and actin. I'm just showing you the microtubules data because it's a bit cleaner. Maybe during the long prolonged questions later, I could show you some of the actin data if you're interested in. And we also really think that intermediate filaments might play a really important role because they're typically responsible for the long-term elastic response. So that's all super interesting. And so Sam, I'll say you'll be happy to know that we'll be going deeper in the mechanics later in this talk, but you'll be unhappy to hear that we're only doing it at the single droplet level so far. We haven't gone to start thinking about the coupling of droplets elastically yet, at least not here. But I have some cool data to show you later about that. All right, so moving on to sort of some simpler, more tractable physics questions. So hopefully, like looking at this biological stuff is inspirational for you, like it is for me. But I don't think I've ever felt like I've really solved a biological problem. I'm much better at solving physics problems. So what I've done here is I'm sort of distilling this down to what I see as the essential physics question. And to me, the essential physics question is, how does a host polymer network affect the phase separation of its solvent? Okay, so that's the simplest way to phrase it. And now it's a physics question. So, and it's schematically shown on the right. So I have a red polymer network, and it is swollen by a green solvent. Now, that green solvent, interestingly, is not a one component solvent, it's a two component solvent. And so now if I quench my system so that the blue and the yellow that make green want to phase separate, they now have to deal with the polymer network as they grow. And if the blue droplets are the minority, if the blue is the minority phase, and if it has a disfavorable interaction with the polymer network, then you can see that as that droplet grows, and you can imagine as that droplet grows, it really has to deform that polymer network. It's going to be dumping energy into that polymer network, and storing energy into that polymer network. And it seems pretty clear that there could be potentially some interesting couplings there that we'll be exploring. And there are interesting couplings that I'll be telling you about today. Okay, so the way the experiments work is we start with something that couldn't be less biological. We start with a silicone gel, okay? We like silicone because it's transparent, and we can tune its modulus from a kilopascal to a megapascal, so we have a huge dynamic range of elasticities. And then we put it in another very non-biological fluid. We put it in a fluorinated oil, okay? That's you would take an alkane and substitute the hydrogens for fluorine. And the thing about fluorinated oil is that it's got a weak solubility in silicone oil and in silicone networks. And so we put it in a bath of silicone oil at an elevated temperature, typically 40 degrees, and we typically do it for a couple of days. And we've made sure, that we wait long enough for everything to sort of reach its equilibrium swelling, which corresponds to a few percent. And then we relatively quickly quench the system, and we vary the quench rate I won't show that today unless you ask about it, but we vary the quench rate. We control the quench rate is the most relevant point to here. And then we watch, because we're quenching so much faster than how things came in, then the driving force to phase separation makes them nucleate and grow droplets of the blue phase on the inside instead of everything sort of just diffusing to the outside when we cool it down. So this is what the experiment looks like. This is a microscope image, well, hardly a microscope image. The scale bar is it's a milliscope image. The scale bar is 100 microns. And you can see very clearly in this image, absolutely nothing. And that makes me very happy because I want my system to be well mixed and in thermodynamic equilibrium. So this is my sample after I've been my polymer network, after I've swollen it for a couple of days in oil. Everything is well mixed and perfectly homogeneous. And then I start quenching the system, and the time step is in the bottom on the upper left. And you can see pretty quickly we nucleate and grow a bunch of droplets. And I see that Eli has a question. Is it Eli or Ellie?
Yes, Ellie.
Sorry.
So I'm confused. Didn't you say that the solvent should be composed of two components?
Yes.
You see the silicone gel and the.
Very good. You're paying attention. So yeah. I have a very funny. I call my solid objects gels, and they're a very funny gel because the mechanically connected network has the same chemical composition as its solvent. So I have PDMS polymers that are cross-linked that give my system, my network elasticity. And they also have PDMS polymers that are not cross-linked, and they're swelling my network. And then when I introduce the fluorinated oil is mixing with the uncross-linked PDMS, okay? And then I push that and those guys separate.
Thanks.
Yep. All right. Okay, so moving forward, so we can make these droplets inside of this elastic network. And one really cool thing about these droplets that we really got for free and that we weren't expecting is that they're very monodisperse. And they don't look particularly monodisperse in a single microscope image because things can be in or out of focus. But if we sort of move the microscope focus up and down and measure the diameter of all these droplets, they're polydispersed to within 10%, which is really nice. Because really I'm making emulsion droplets, uniform emulsion droplets that are highly monodispersed. This is actually really hard to do. People normally make them one at a time in a microfluidic chamber, right? And here we have a bulk thermodynamic approach to making emulsion droplets. Of course, because Sam is in the audience, I have to say, of course you can do this at the nanometer scale with, AOT and all those cool surfactant-y sort of things. But at this micron scale, there's not another way that I know of that you can make these emulsion droplets so efficiently in the bulk. And if you don't like real space, I'll convince you in Fourier space. If you take a laser pointer and you shine it through this sample, you get a nice ring of a diffraction pattern telling you that you have a dominant length scale. And if you analyze it, you find that that length scale in the ring comes from the form factor, not the structure factor. So it's really saying that these things are monodispersed droplets. Okay, so if we do this phase separation process in an elastic network, we tend to get something monodisperse, and they don't coalesce, which is also really cool. But I'm not going to dwell on the emulsion physics here. But I mentioned that, you know, the elasticity is what seems to be making it monodisperse. And so maybe it comes to no surprise to you that the magnitude of the stiffness determines the size of the droplet, okay? So softer networks have bigger droplets than stiffer networks. And here are the size distributions. So we have a few different curves here. The bright cyan curve corresponds to our stiffest network at 850 kilopascal with a pretty tight size distribution around 5 microns. And as we make the system softer and softer, you can see that the droplets get bigger and bigger. The width of the distribution increases just a little bit as it expands, but as a percentage, the standard deviation of the distribution is roughly constant compared to the mean, even though the mean is shifting a lot.
Eric, I have a quick question.
Super.
So this is with changing elastic stiffness, but what about the pore size of the network? How does that change?
Yeah, that's a fabulous question. So there's two things to say about it, right? You know, we know from classical polymer physics that, you know, as the thing that sets the scale of the modulus is basically the scale of the pore, the correlation length of the mesh size. And so as I'm making these things stiffer, indeed, of course, I'm making the mesh size smaller. But on the other hand, what's incredibly cool, and the fact this is the thing that makes it completely different from the motivating data I showed you inside of cells, is that that mesh scale in the polymer is at the nanometer scale, where these droplets are on the micron scale. So we really have a separation of length scales. And I argue that these droplets, by the time I can see them in my microscope, are really interacting with a continuum, not with a bunch of discrete filaments. Whereas in the cells, you could see that, you know, that there was a completely different situation where the droplet is on the scale of the mesh. So that's an important thing to remember.
Okay, that's very useful. Thank you. Yeah. And as the droplets, and presumably you will explain this in terms of what selects the size of the final drops, and will flow, do you expect poroelastic flow to arrest the size, or is it just going to be elasticity?
So yeah, well, let's revisit that at the end, Suraj. I mean, it's really the $1 million question. And what I'll tell you right now is, honestly, there's a bunch of different theories about why the droplets are all the same size, and I don't believe any of them yet. So they're all very plausible, but they're not consistent with my data. So the jury's out. That's the preview, but we can go back into it.
Sounds good. Thank you.
Yep.
So is the total volume of the droplet the same? Because it seems in the picture that you show that there are more droplets when they are tiny.
Super. Excellent, Natalie. Yeah, good point. So to the best of our ability, you know, we're doing light microscopy. But to our best of our ability, the total volume of the liquid phase that we make when we cool is unchanged when we quench by the same temperature difference. And the physics underlying that is that it turns out, and we can discuss it more if people want at the end, is that for interesting reasons, the solubility of the oil inside of the network in the first place doesn't depend on the stiffness of the network. So for all these stiffnesses, the amount of oil that we get in the first place is independent of stiffness. And then we cool it, the amount of stuff we get out is independent of the stiffness. It only depends on the temperature. Okay, so moving on. Yeah, so we're trying to push this down to smaller length scales. A big theme in my lab, which I really won't have time to talk about today unless people ask about it at the end, is we're interested in processes that living organisms use to make structural colors. So typically what they do is they have a phase separation process that they control precisely at the nanoscale to make much smaller droplets than I'm showing you here today. And Alba's a PhD student in my lab, and she has some really nice results that we'll be putting up on the archive soon, showing that if we move out of PDMS, which has a maximum modulus of around a megapascal, and switch to polystyrene, we can actually get droplets at the nanoscale, and those droplets can produce color. Anyway, so watch out for that in the archive, and if you ask about it at the end, I can show you more of her data. So yeah, okay, but still, I haven't told you what's the underlying physics, okay? So to get at that, what I'm going to try to do is I'm not going to just tell you the answer. I'm going to try to, show you a bunch of data and help you get there on your own. And the first thing you might think is that the nucleation process is very different for these droplets in the different stiffness networks. As Roy asked, because the microenvironment is very different at different stiffnesses. And so this is what our data looks like. This is what the nucleation process looks like at different stiffnesses. We've got 4 different samples here. They're increasing in stiffness in the counterclockwise direction. And you can see that the soft samples will start growing, nucleating and growing at relatively small amounts of undercooling. But as we get stiffer and stiffer networks, we have to cool the system more and more below the temperature where we equilibrated it with the fluid, okay? And we can quantify that. On the left, we have a plot of just the temperature at which we observe nucleation versus the stiffness of the network on a logarithmic scale. 40 is the temperature at which we equilibrated to the bath of the fluid. And yeah, so for the soft stuff, we need to go less than a degree of undercooling. We're cooling pretty slowly, I need to point out here, right? So we tested the rate dependence here. We appear to be in some sort of, in the limit where the nucleation temperature is very weakly dependent on the quench rate. Yeah, but by the time we get to stiff things, we might have to undercool about 6 degrees or so before we see any droplets. Okay, so
Eric, Eric, sorry, what are the squares that we see in the video over there? There's like
the squares. Yeah, that's just bad imaging, and that's the reason is that I use microscopes because I grew up as a colloid physicist, and microscopes are designed to image micron size objects, and I'm imaging these giant objects with a very, very low magnification lens. So the illumination, those squares are just basically like problems with my illumination. Yeah. So there's no physics of those squares. Yeah, that's just bad illumination. So I'm using a microscope outside of the parameter of what it was designed to be used for. So great observations. Okay, so nucleation is very different. Okay, so the obvious hypothesis, right? And we should always start with the obvious one, is that somehow the elasticity of the network is creating a barrier to nucleation that fits into the framework of classical nucleation theory, which we all love, even though we know it doesn't work anyway. It sort of works, but anyway. So let me show you some data to convince you that it's absolutely not a barrier to nucleation. This is a beautiful experiment that Tsai conceptualized and performed and analyzed. So what she does is she takes a sample, a stiff sample, she equilibrates it at 40 degrees C for a couple of days, and then she starts cooling it down in the light microscope at a quench rate of 0.2 degrees C per minute. And then she just measures how much particle do I see, right? So she just basically counted a number of pixels that were above some threshold from, the straight transmission. And you can see that she was able to quench it all the way down to 7 degrees below the original temperature, 7 degrees of undercooling before anything happened. And then suddenly, we started really growing a lot of droplets. Okay, this is completely, so far what I've shown you is completely consistent with having a strong barrier to nucleation. However, the really cool thing happens is when she just heated the sample up by 2 degrees Celsius, okay, still well below the temperature where things were saturated, and the droplets all dissolved. Okay, so these droplets are dissolving into a network that is super saturated with a liquid. Okay, so they're super saturated with their components. So this is something different. It looks thermodynamic because you can cycle the temperature and it comes back and it disappears again. Okay, and so so this is not a nucleation barrier. What the heck is going on? So we're gonna start thinking about the mechanics, and for the rest of the talk, we're gonna be jumping back and forth between mechanics and thermodynamics, okay? To try to understand what I'm showing you. The first, mechanical fact, ground zero here is what the heck is happening when one of these droplets grows? Like how is the polymer network, what are the constraints due to droplet growth? Here we have this image that's taken with a Raman microscope, a fancy Raman microscope tuned to the vibrations of silicone. And what you can see is that the droplet itself has no traces of any silicone inside of it. And we have a pretty homogeneous distribution of silicone outside of the droplet. So this says as the droplet grows, it really does have to exclude the polymer network, okay? So that's like mechanical truth zero. And now if we sort of put together a toy model that we'll come back to in a little bit more detail later, sort of imagining how a network would have to deform when you have such dramatic expansion in it, this is a simple analytical model where we assume the network is incompressible. I'll describe how you derive it in 2 lines later. But you can see, that this network, this droplet, which is only increasing by a factor of 70 in size compared to the mesh scale, already has to make huge deformations to the network. My droplets grow by a factor of 1,000. So their deformations are inconceivably large, I would argue, okay? So there are lots of different things that might happen. You might imagine that this network would break, that it would crack, that it would do all sorts of things, and we'll get back to breakage and cracking later, but it certainly has to push. At a bare minimum, it's gotta push against that network to deform it that much, okay? And so the second law of thermodynamics tells us that since the droplet growth is doing work on the network, there's a limit on how much work we can do because it's fueled by the phase transition, right? So the first line here is the second law of thermodynamics at constant temperature and pressure, okay? It says that the work done by the droplet to grow has to be less than its the change in the free energy of the molecules that are making up the droplet in the process of condensation. The work is easy to calculate, thank God. Mechanics is hard, but since my droplet is just a liquid, it has a trivial state of stress, right? So it just has a pressure. So the work that the droplet does is PdV. And then the free energy liberated by condensation is just the difference in the chemical potential of a molecule in the droplet phase versus a molecule in the dilute phase outside, okay? And if you plug in, sort of the ideal expression for an ideal solution in the dilute phase, you get this critical thermodynamic idea that there's a maximum pressure that the droplet can exert while growing. We call that the condensation pressure or the stall pressure. I might use both today. And it's got 2 parts. It's got a factor in front that's NKT, where N is the density of molecules in the liquid phase. And in this experiment, that prefactor has a magnitude of around 10 megapascal, which is about an order of magnitude higher than the stiffest networks that I ever use in these experiments. And then there's a logarithmic factor that depends on the concentration of the molecules in the dilute phase relative to their saturation concentration. So if the molecules are at their saturation concentration, they can't do any work, they can't exert any pressure. If you have a super saturation, they can push, okay? So that's the basic thermodynamic concept. So let's see if we can use that, okay? And apply it to the experiment I just showed you a moment ago. So the key thing is that as we cool the system, right, the saturation temperature, the saturation concentration changes. So, but Tsat is the temperature at which I equilibrated. So if I haven't made droplets yet, my continuous phase has this concentration psi Tsat. And as I cool, this factor is dropping and dropping and dropping. So what happens is that condensation pressure starts off at 0 at the equilibration temperature, and it keeps ramping up as we cool and we cool. And then what we see is that sort of these events of where droplets appear and where they dissolve all happens in a regime around approximately 800 kilopascal, which is very close to the elastic modulus of this material. Okay, so it seems that droplets seem to form when they exert a stress that's of the same order of magnitude as Young's modulus for that material. But we're going to go deeper than just leaving at that. We're going to try to quantify things a bit more. So we're going to go back to this nucleation experiment. And what we're going to do is we can use we have a phase diagram for our system that I'm not showing. I don't want to bore you. But we can basically calculate this condensation pressure at the point of nucleation for our system. We know that we have a certain amount of undercooling. With the help of the phase diagram, we can translate that into a pressure. And what we see is that the condensation pressure that you need to make a droplet scales with the modulus, okay? So it makes sense. The stiffer the material, the harder you have to push. Weirdly, and I would even go far to say as unfortunately, that scaling is not linear in the modulus. I don't know why yet. I have some ideas that we'll allude to later, but it seems to be a little bit more complicated than just a simple scaling with the modulus. But what I want to do now is I want to tell you about sort of the classical mechanical way that you would approach this using standard solid mechanics. And this is a really cool calculation that I'd like to outline for you, because as a physicist, I was always afraid of continuum mechanics because there are all those equations and different partial differential equations, but it's really surprising how much you can do without any partial differential equations. And this is a really nice example. So I want to tell you about the classical cavitation theory of Ghent and Lindley from 1959. The basic idea is I start off with some cavity in an empty thing in a solid material with a pressure 0 inside and some initial radius capital R0. And I'm going to keep track of some material point inside of the matrix at some distance little R0 from the center. Okay, so initially my system has P equals 0. Then I inflate, I increase the pressure. The radius of the cavity increases, and this material point I'm following gets pushed out. Okay, now if the matrix is incompressible, okay, it's really easy to calculate the kinematics, because you have to conserve the volume of the matrix between this material point and the boundaries of your hole. Okay, so I can just write down that, you know, the volume of this spherical annulus, the spherical shell in the initial state has to be equal to the volume of the spherical shell later, and this gives me this relationship between the final position of any material point in my sample and its initial position. And I used that to make that animation earlier. Now the thing is, how do we use that to calculate what's going to happen? We need energy. Okay, and the basic idea here is that, you know, we have that the amount of work that we have to do in the system is equal to the amount of energy we store in it. Okay, so this is zero temperature calculation. Now in elasticity, what you say is that there's some energy density W, and that energy density W depends on where you are in the sample and how much each of those material points have moved. Okay, now in small deformation elasticity theory, which a lot of you may have seen, or most of you have seen probably, that the energy density has a really simple form. It goes like the elastic modulus times basically the square of the strain matrix, okay, epsilon ij, epsilon ij. This is easy to calculate once you know all the displacements. And then what you can do is you can just basically minimize an energy, find what radius of the hole minimizes the energy for each pressure, and you can calculate the relationship between radius and pressure. Okay. Now the amazing, crazy thing is, is that there's an instability. Okay. At small pressures, it doesn't do much. You know, the radius of your cavity increases linearly with the applied pressure. But when you get to a critical pressure, which people call the cavitation pressure, you have this cavitation instability where this droplet this cavity can grow forever at a fixed pressure. Okay, so that's this magic instability that happens that Ghent and Lindley described. So there's lots of reasons to criticize this, primarily because in my system I have very, very large deformations, but let's just very irresponsibly apply it to my data anyway. And what we see, so if we say, okay, if we say my droplets nucleate when they have a pressure that exceeds this cavitation pressure, it should follow this white line here. And you can see that doesn't do very well for the softer matrices, but it does seem to do pretty well for the stiffer ones. So it might be some sort of lower bound, effective lower bound on the condensation pressure. But we can move ahead with this though. Even though we know it doesn't fit our data perfectly, and later we'll discuss why it doesn't fit the data perfectly if people want me to, we can just run with it and ask, what does this mean for the thermodynamics? So what I want to show you here is this very simple phase diagram that we proposed last year, putting together the elasticity and the thermodynamics with these concepts. This is an isothermal phase diagram. Usually for phase separation, you have composition on one axis and temperature on the other. Here we have composition versus elasticity. And so here, on the left, we're at zero modulus. Then you expect a direct transition from a mixed state to a phase separated state. That corresponds to classical liquid-liquid phase separation. But if you have finite elasticity, what our thermodynamic analysis is telling us is there's this region of the phase diagram that we're calling stably supersaturated, where you couldn't put any more monomer in if you wanted to, but it can't phase separate because it doesn't have enough pressure to overcome this cavitation instability. And this actually does a pretty good job in describing our data. So our experimental data had temperature versus modulus, and I can sort of flip this phase diagram over. If I assume that the critical pressure is just this classical 5E/6, it gives me this boundary between phase separated and stably supersaturated, and it sort of mirrors what we see in our experiment. Typically in our experiments, we have to undercool even more still than we expect, okay? There's lots of reasons why we might have to undercool more. It could just be the residual, I would say, nucleation effects. Like you still have to overcome a nucleation barrier from surface tension. That might be why you have to undercool a little bit more. Or it could be also some limitations of our mechanical model. To make this, I needed to use a model that was assuming basically small deformations in a system that clearly has very, very large ones. Okay. So I think what I'm going to do now is, because we've actually already used almost an hour, is I'm going to actually just skip the next section of my talk, which digs in deeper on the mechanics of this stuff, give you a summary, and then we can go back to the mechanics or focus on whatever is most interesting to you all. So jumping to my summary slide. Yeah, so when we have a process of liquid-liquid phase separation in an elastic network, things are very, very surprisingly different than they are in classical liquid phase separation. First of all, it's very easy to make uniform droplets. Their size depends on the stiffness of the network. The compressive stresses from the network stabilize these supersaturated mixtures. That was the last point I just made to you about the thermodynamics. Then now here's a few points that I haven't yet justified for you that we can go back and discuss. That the network stresses define the droplet shape. Normally we think of surface tension making a droplet spherical, but in our case, it's actually elastic forces that seem to be doing it. Another important thing, which probably will come as no surprise to you, is that as these droplets grow, elasticity fails, and specifically we do damage to our network. And the last thing is that the ripening process looks very different from classical Oswald ripening. And instead of droplets material moving down from small droplets to big, material tends to move down stiffness gradients instead of from small droplets to big gradients. And we're trying to make connections between cells and gels, and we love talking to theorists. So I'm going to stop here, and then we can jump around however you all would like.
Thanks, Eric.
Mm-hmm.
Okay.
So many questions during the talk, but none at the end.
I actually have a question. Okay, yeah, Michael.
I was just thinking about this growth into a gel. The gel has a real elasticity. If you think of growing in a cytoskeletal network in a cell or in some kind of a polymer melt, or something which doesn't really have a zero frequency
Yes.
elasticity.
Exactly. So that's an essential point. And I would say this is something that it's been explored theoretically. That has been explored quite extensively theoretically by Hajime Tanaka. Starting in the 90s, he had a process he called viscoelastic phase separation. And there, what he was thinking about, basically two fluids that were phase separating, but where one of them was viscoelastic that would have some sort of relaxation. So in that case, there are all sorts of really interesting phenomena that arise. Most of Tanaka's work, though, is focused on things related to spinodal decomposition. So it's mostly in a different type of section of the phase diagram than what I've been telling you about today. I'd be curious to know what happens when you're more in the nucleation and growth regime that I've been describing. But if I needed to sort of just recklessly speculate, or I will now recklessly speculate, that as long as sort of the characteristic timescale for growth is shorter than the elastic relaxation time of your polymer network, that you would probably see pretty similar things with uniform distributions. But I expect it would get pretty interesting when the kinetics get comparable to each other.
I'm muted, so thanks.
Yep.
If I may, what happens if you try to take a given experiment and then after you see the location of the droplets, dissolve them and try to reconstitute them in terms of the pair correlations of where they show up?
Super. That's excellent. So I have not done that. I'll answer that with two bits of data. One bit of data I'll tell you about, and another a plot I can show you. So the first experiment is the first thing I want to tell you is more closely related to your question. So what we see when we heat it back in to redissolve and cool it again, about half of the droplets appear in places where they were before, and the other half appear in some new location. And I say about half, I mean, that's what my eyes say, that's not what the computer counted. And we haven't done that experiment systematically. We haven't cycled a bunch of times. But if we're going to talk about pair correlations and locations, we do have, and this might also connect to Sam's comments about Eshelby theory potentially, that we can measure the pair correlation function in 3D for our droplets after we've made them. And what we see is that there's a very strong anti-correlation in the locations of the droplets. So so here we plot G of R, and this R equals 40 microns is the average droplet spacing. And once you get past the average droplet spacing, there's no correlation in the droplet locations. But as we get closer and closer, there seems to be a more and more pronounced repulsion. And again, you should be super careful. This is an actual G of R now between two objects, but you need to be careful how you interpret it, because these objects are not free. They have no translational degrees of freedom. So this looks like a repulsion. It doesn't mean they're pushed away from each other. It just means that somehow the nucleation process prevents them, or the growth process prevents them from being found near each other at the end.
This is a snapshot in time.
This is at the end when it's all completely done growing. We've not done it in a time resolved way.
Can I interrupt? Could it be,
yes, of course,
could it be a depletion of the concentration in the solution in between?
Yeah. So there's two possibilities here, right? So there's two possibilities for this is clearly a long range interaction, and there are two long range fields, at least two that I know of in my problem. One is the concentration field of the stuff that's condensing to make a droplet. And you would expect that if there's a sink at one location, there'd be a very slow, like 1/R type decay in the concentration field. That might be the reason that the growth of one droplet inhibits the nucleation of another. Another possibility is that it could actually be the elasticity, right? So the elasticity is also long ranged. You're deforming the network around you, and you're going to have some, with some functional form, that deformation is going to fall off. And if the growth, we saw everything about the droplets was coupled to the elasticity. If it's more generally coupled to the stress, that might be another way that we get this pair correlation.
Eric, one way maybe to check it out is to do it at different concentrations of the of the little particles, right? Because if you do them at a very low density in comparison, then you will be able to differentiate between those two options.
Yeah, you know what? We only did it with the when we had small droplets that were close to each other, just because we needed to get good statistics with our paracorrelation function. So we didn't. This was really the only this situation with the smallest droplets closest to each other was the only one where we had good enough statistics. And I saw that Sam had a question, and since he asked about Eshelby, I think he might want to ask another question now.
Actually, yeah, I did want to ask something. In the chat, I've been going back and forth with Asi Zemel from Hebrew University, who also brought this up. In the 70s, when even I was a little boy, there was a lot of work on phase separation of hydrogen impurities in metals. And metal is an elastic medium. The hydrogen impurities would aggregate. And I'm not aware of the studies there, the nucleation and growth, but you might want to look back if those ancient papers are written on papyrus or something, and see if people had looked at the nucleation growth of the hydrogen droplets in metals. I know that there was a guy, Zabel, at Urbana, who looked at the very late stages and showed that there were interesting elastic effects, but there might be some interesting analogies.
Thank you. That's super helpful. Yeah, there's a lot. I mean, what we found in this problem, there's often lots of really interesting connections with the
So this one is not just me. This is Eric on offline.
Oh,
Okay. So Effie, maybe you can continue.
No, I was just going to say to Sam that I think this is coming back with the hydrogen storage, that storing hydrogen in metal lattices, in metal frameworks of some kinds.
that was one motivation in the 70s. And the other motivation in the 70s was that the hydrogen impurities caused the cracking of, I don't know what, titanium covers of nuclear reactors, and which you don't want to happen. And people didn't understand why they were cracking until they realized that the hydrogen was phase separating. So you had one part of the metal that was expanded by the hydrogen impurities and another part that wasn't, and there were cracks between them. But I mean, the metals are not soft elastic gels, and I'm aware, and Asi, who brought this up originally, is aware, but it may be interesting to look at the analogies, and there is a literature.
That's super. Thank you very much. I had some technical difficulties, and now it's.
Hi, Eric. Oh, welcome back.
I'm now on a different. Now I'm on a different computer. I might. Yeah. Give me a moment to sort of deal with the fact that I needed to switch computers. Okay, and now I'm I should be completely back.
Eric?
Yes.
Yeah, so two questions.
Yeah.
First, the technical one. You're probably the last plot which you have shown was the temperature where the droplets form as a function of the modulus. And there was a mismatch between the theory and the experiment, which you suggested may be related to remnants of nucleation problems.
Mm-hmm, exactly.
If it is so, then I mean, that's a simple experiment to do. You can just go other way around, just heat the droplets and measure the, which you did, and plot on the same plot the temperatures of their dissolving, right?
Exactly. Yeah. So that's an excellent experiment. It's one we want to revisit. But you're absolutely right. We should be able to resolve that question by systematically doing that one-off experiment that I showed you where we just did a discrete step by 2 degrees, but it's maybe slowly heating it up and then observing where it dissolves. That's a great suggestion. Thank you.
And also when you say that these compressive stresses, I mean, your model suggests that the compressive stresses stabilize the supersaturation. In a sense, that's only on the level of the continuum theory.
Yes.
I would expect that on a nanometer scale, you should still have detectable.
Exactly.
Which I wonder whether you can see them maybe by dynamic light scattering, or I mean, they are really small but still probably detectable, right?
Yeah, exactly. So there's a lot to say about that. Yeah, we call that the baby droplet hypothesis, and I think it's really interesting. And one way to say it is that what I'm calling nucleation isn't really nucleation. Maybe nucleation is happening at a much smaller scale that I never resolve, and that there are countless number of unresolved baby droplets that I don't see. And then what I'm actually calling nucleation is really some growth instability of all these baby droplets. So yeah, that's a really interesting idea. And I know I've been speaking with Pierre Ronceret, who was a postdoc at Princeton, who's now starting his own lab in Marseille, as well as David Zwicker, and his postdoc Estefania in Göttingen. And both of them have been really trying to develop some theories that go from the structural scale of the polymer up. And both of them propose that what your hypothesis makes a lot of sort of thermodynamic sense. It doesn't always work that way according to their theories, but in certain parts of perimeter space, it makes a lot of sense. And for me, actually, a year and a half ago, I wrote a grant to get an X-ray scattering machine in my institute so I can probe this exact problem. And that grant was funded, and I hope that by this summer we'll have the machine installed, and we're precisely going to be looking for these nanodroplets that you suggest.
Aren't these nanodroplets detectable even by DLS? I mean, if they're
they don't move. Right? I mean, they're not super dense, right? So they're pretty dilute. I mean, well, actually, I don't know what the density of the nanodroplets are, but they don't move, right? So they might be a little bit of cloudiness. So if we tried to do measure a difference in the scattering, maybe if we had a really thick sample, we might be able to see a difference in the turbidity. Yeah, we've never tried with DLS, so I always assumed it'd be too hard, but maybe we could do it. So.
X-ray is better. Yeah. Oh, X-ray is better. That's
X-ray is definitely better if you have the.
Yeah, but I still don't have an X-ray machine in my lab, right? And
I can help you. I have one. So, Eric, it's okay. We can.
Send me an email. We'll send you a sample.
All right, sure.
So I have a question.
Yes.
So in the beginning, you showed some pictures in droplets in the cell. So and those droplets were not same in size. So is that not a steady state, or is that?
Super question. Yeah. So that's that there's lots of potential reasons for that. One, the most skeptical answer is that my other experiments have nothing to do with what's happening in the cell at all. I admit that might be a possibility, but maybe a more generous interpretation, assuming that they're related, is that it could be is really all about this issue that for the droplets in the cell, they're really interacting with the polymer network at the mesh scale. And polymer networks are very heterogeneous naturally at the mesh scale. So if I'm looking at droplets at the mesh scale, like the ones in the last question, or these that I see in the cell, I would generically expect them actually to be pretty heterogeneous. The material only becomes homogeneous when you get to the continuum scale, right? So
because of the pressure and the cavitation instability.
Yeah, and also, you could not necessarily. I was trying to make a simpler point. The simpler point is that if I take a slab of a polymer network and I describe its structure statistically at the mesh scale, there's a huge amount of variance. But if I zoom out and I coarse grain and I look at a larger volume, there's much less variation in volume to from point to point in a material when you're looking at larger scales, right? So the concept of Young's modulus makes a ton of sense at the micron scale, but it doesn't make any sense at all at the nanometer scale.
So I think the microenvironment is different for each droplet. So if I think about the physics of a single droplet, then I can still give a theory, and that will work in the cell. I mean, we can hope.
Yes, I think that's, yeah, I think so. I especially if you account for heterogeneities. And, for us, we're doing this work with, Tom is doing this work with microtubules in my lab. And, what we really want to get to a point where we can actually even measure how microtubules are deformed by these droplets, especially if we move to super resolution methods. And there, I think we'll really be able to make, significant statistically significant statements about the local environment of a droplet and relate it to its growth.
I see two hands, just Matilda and Matan have been waiting for a while.
Yeah, thank you. Eric, I have a question related to your blue colored sample.
Yeah.
So if I understand well, the droplet composition is perfluorinated oil, right?
Oh, it is, but not for that experiment. So in the experiments where I had the blue sample, it was a completely different system. And there the droplets are actually not droplets, they're particles, and it's PMMA inside of polystyrene. So in that blue sample you were seeing that looked maybe something like this, the one that I showed during the talk, microscopically it looks like this thing I'm highlighting here, where each of those little white dots is basically a PMMA particle. And they're not nearly as uniform as we were seeing macroscopically. And we don't know if that's because sort of the polymer network is inherently more heterogeneous at that scale, or we just don't have enough control about how we're quenching the process. And we're quenching it in a very, very different way than we quenched it in the rest of the experiments. And the rest of the experiments, I started with a homogeneous system and I quenched it by cooling it. The way Alba does it is Alba does everything isothermally. So she swells a polystyrene melt with MMA monomer, an initiator, and then she heats it up. And at first it stays well mixed, and then that MMA starts to polymerize. And as the MMA polymerizes, its solubility is dropped. And then that MMA grows a little inclusion inside of the polystyrene matrix. So there's a lot about the physics in that example that makes these blue samples that makes it very different. But also I think fundamentally there are deep connections because we are trying to grow something inside of a network with some elasticity.
And were you able to make very different colors?
Not yet.
Right, for example?
No, not yet. Because the problem is that to get red and anything, this beautiful blue color is the color of the sky, right? So to get the color of the sky, all you need is really small objects that have a significant scattering cross section but small objects smaller than the wavelength of light, but scatter well and are uncorrelated. And that allows us to get, that's all you need for a blue color. For us, we actually can get some correlation. So this is X-ray scattering data at a completely different length scale that we did at a facility in Switzerland, the Paul Scherrer Institute. And there we actually see that we do have some structural correlations in this material, but they're not strong enough structural correlations to make vivid colors. Birds, however, can make colors, all different colors, not just blue, by a similar process with strong structural correlations. And that's something we're trying to get to in the long term. And the paper describing this stuff I'm telling you about now will be submitted to the archive tonight. So it should come out the day after tomorrow, I'm guessing, if you're curious about that.
And is it, in a last question, is it possible to make a bulk sample, or is it just a quite thin one?
Yeah, so right now, so what's interesting is in the process that we use, what's cool is we swell the whole sample in bulk. We drive polymerization throughout the whole sample, but the polymerization process is a little bit different on the surface than on the bulk. So most of the color producing stuff does sit on the surface. And you can see that, especially, you can sort of see it in this picture and see that from the side, it just looks like white, the top is more blue. So yeah, most of the color producing stuff is confined to something about 100 microns thick.
Okay, thank you.
Yeah, thank you.
Matan?
Yes, thanks, Afi. Eric, great talk. I have a question for the formation and actually the steady state of these droplets. And I guess you looked at many of these videos already. So first, it looks like not only they're non-dispersed, but they also all appear at the same time, at the same size. So it seems like they're very, very strongly correlated. One thing that you mentioned is that they're correlated through elasticity. But I was wondering, yes, exactly. Also, there's a typical size for their separation. Are they also correlated through flow? So if you dye one of these droplets after they formed, does the dye go to other droplets or it stays there?
That's a great question.
I mean, dye in a sense that use without changing the material properties.
That's very nice. Yeah. So we haven't done it with a dye, but I'll show you some data now that shows that there is communication through the monomer, through the condensed phase. You can transport material and exchange material between droplets. And that was in part of the talk that I skipped, where we discussed the coursing behavior of the droplets. And so the basic experiment, I'll just tell you really quick, is we make the polymer with a stiffness gradient, and we swell it on both sides, and then we grow. You know, when we cool it down, we get small droplets on the soft side and big droplets on the small droplets on the stiff side, large droplets on the soft side, and then we just look and see how the system ripens. And I'm going to switch to presenter mode now. So far away, you can see this is the soft side on the left. This is the stiff side on the right. And in the middle, you can see the interface between the sharp and the stiff sides. And this is right after we've sort of nucleated and grew the droplets pretty quickly. So you can see this interface is pretty sharp. And you know, if we just let the system relax, I'll just pause the movie, sorry. Yeah, so what we see, we can see the small droplets shrinking and the large droplets at the interface growing. So there is the possibility to couple these things with flow as you asked. And the way what we think is going on here is that basically the pressure inside of the small droplets is bigger than the pressure in the big droplets. And normally you would say that's true because of surface tension. We think it's true because of this cavitation, because of the elasticity and the continuum mechanics. And the way I'll prove that to you is what I'm going to do is I'm going to reverse. I'm going to reverse the stiffness gradient. So what I have here is I have a system that's 140 kilopascal with one type of PDMS and big droplets, and I have a softer PDMS made from a different company that produces monodispersed droplets that tend to be a little smaller. And if it was classical Ostwald ripening, the droplets on the right would shrink to feed the droplets on the left. But here you can see on the left, these big droplets shrink and feed the growth of the smaller droplets on the right. Okay, so in some ways this is just like Ostwald ripening because there's transport in the dilute phase of monomer driven by pressure. But the key thing is that the pressure we think is not set by surface tension like it would be in a canonical Ostwald ripening process. It's set. There's a contribution from the surface tension, but there's this extra pressure on the inside that is the network squeezing against the droplet. And we think that's what drives the transport over. over a long time.
That's really cool. Thanks.
Yeah.
And Eric, I just maybe just continuing on this question with regard to dynamics.
Yeah.
I wanted to go back to what I had asked earlier with regard to flow. And so can you now modulate along with an elasticity, elastic modulus gradient? Can you also have a porosity gradient? And do you see different phenomena, or do you expect to see different phenomena?
Yeah, that's interesting. I mean, very sort of naively, I mean, unfortunately, the two are so strongly coupled experimentally that I can't manipulate them, right? I mean, if I change the stiffness of the material, it's going to be hard to do that without changing the permeability, and vice versa, right?
Sure. Yeah.
But by using like in the last slide, when I use PDMS like from different companies. That's my naive physics way of thinking about it. But if I have more of a chemist to say, well, if I use different elastomers that have different network topologies, maybe I could control those separately. So I think it's something that is hard to do, but could be done. In terms of what would I expect, I mean, certainly, if I take a thermodynamic view of this, it wouldn't make any difference at all.
Sure, but it's a question of dynamics, yes.
Yeah, and then no, it might be a good way of separating the thermodynamics from the dynamics.
You might have interesting ways of patterning different sized drops in different regions by playing with these two, playing with porosity and elasticity. The second sort of question, again on the same note, was to do with sort of the motivation that you had, which was in the cellular context in the cytoskeleton, where you don't have as much of a scale separation as you do in your setup, in your experiments. And in that case, one might expect, because as the droplet is growing, given in addition to the cavitation pressure threshold, if as you're compressing the network and the mesh on the outside of the droplet and you're excluding it, and so presumably you can also you might also have smaller pore sizes right outside the drop.
Yes.
And it increases further away, and so presumably there will be a slowdown of the growth, not just from the elastic pressure, but also because fluid can't get out, because the pores get smaller and smaller as the mesh gets compressed.
Yes, and no, that's an excellent point, actually. And so there's a few things and I thought about those questions more in the context of the polymers than in the cell. I think in the cells, the filament networks are still so dilute that I don't think they would really seal things off very much, and also because the strains are very low in the cell. Like, we might open up a pore that's a few times the mesh scale, not 1,000 times the mesh scale. So one idea that Rob has been thinking about a lot is that when you grow, these things to a factor of 1,000 and you're stretching all these chains right next to each other, they're being stretched, they're being aligned. It wouldn't be surprising if you could even crystallize chains at the surface, and then that would completely kill any transport of stuff on the inside and the outside. So I think that's a really cool idea that I think is related to what you were suggesting, that we haven't probed it yet to see if there's any signatures of crystallization. What we've looked for is birefringence, and the alignment, such strong alignment will give you sort of on a per unit volume basis a huge birefringence. But unfortunately, these samples are so small, these droplets are just a few microns, it's hard to measure the birefringence on those scales. And there are some instruments that can do it. We don't have them in our lab, where we've been flirting with people to get access to them.
Okay.
Yeah, so I think birefringence would be the way, might be the thing to look for there.
Yeah. Thank you.
Thanks.
Sam?
Sam, do you still have your hand up on purpose?
So if I may, did you try shearing your samples or compressing them uniaxially?
Great question. Yes, we have, and we haven't done shear. We've done some compression in a geometry of like sort of a thin film geometry. We're compressing the whole thing. There, what we were trying to do is to try to see if we could get some mass transport down a stress gradient explicitly, because I've shown you mass transport down the elasticity gradient. It'd be great to prove that that's stress transport down a stress gradient specifically, because that would be a much more general and beautiful way of manipulating these things. You could poke here and make it appear somewhere else. Those experiments, I would say, have been inconclusive so far. There's a number of experimental subtleties. So far, all of our experiments, we've squished them, we've seen the shape change, but we haven't so far seen any coupling to dissolution. But I haven't given up hope yet.
Thanks.
Mm-hmm.
Do you know of a way of using this method to prepare alkane emulsions? In particular, is there any way to destroy the elastic network later? Oh,
super. Yeah, no, I'm sure you could do that. We haven't done it yet. If you're interested in trying to do that, I'd be happy to discuss with you offline some ideas. The key thing is that you don't want to use a chemically cross-linked polymer like we're using now. You're going to want to use a network that has maybe a thermo-reversible cross-linking or a photo-reversible cross-linking. And the other thing too is that, it can be, you get to get these, to sort of see the things that we see, you need to sort of be in a sweet spot in the phase diagram, right? So you need to have something that is not like super soluble in your polymer network, and you have to have something that has a significant solubility. So you need to work in a region of around 5 to 10% solubility. And so if you can find, a polymer network that gives you 5 to 10% solubility of your alkane in it, and you have a way of reversing those crosslinks, then I think it should totally work. That's the principles that I would use. I'd be happy to discuss specific options with you offline some other time.
Okay, thank you.
Eric, I'm a little bit lost. So you're saying that if you have a reversible cross-linking, this phenomena will still persist? I thought that would like destroy the entire idea of, because it will just deform over there, cross-link someplace else, and then you wouldn't have this elastic deformation battle to the phase separation.
So you can destroy it. You can heat up or this. I mean, there should be a separate button for destroying the network.
We're not talking about sort of something where the crosslinks are breaking and reforming naturally under fixed circumstances. We're talking about, I have a network that is covalently cross-linked. I grow my droplets. I've got all these beautiful droplets. I want to use them outside of my network. How do I get them out? If you had a way of breaking those cross-links, either by changing the temperature or doing something else, that might be very useful for making uniform emulsion droplets.
But if you have like a chemical cross-link that can reform spontaneously, break and reform, wouldn't you then the entire homogeneity of the droplets and so on should die off, right? I mean, that's the logic.
Yes and no. I mean, this is related to Michael Elbaum's question. I think it was Michael that asked at the beginning that that if my answer is that if you can grow those droplets to the point where they're all the same size faster than the characteristic time scale that these bonds are being broken and made, then I think you could still make a uniform emulsion, right? But certainly if the growth process is slow compared to the time scale of those bonds being broken and remade, then I completely agree with you that all of the physics I described today would completely go away.
Okay, so
thank you so much.
No problem.
Eric.
Yeah.
Thank you so much for that. talk and for that vivid discussion. And hope you enjoyed as much as we have.
Yeah, it was great. It was super fun. So yeah, I'm happy to, I'm happy to stick around and talk longer for anyone that has specific questions. But the rest of you should certainly feel free to leave at any point.
Yeah. All right. Thank you, Eric. Thank you so much.
Thank you very much.
Thank you.
Thank you.
Thank you.
Hey, Eric, just a really stupid comment.
Yeah.
You could, in your elastic medium, if you put a small crack, you could get huge stress gradients, and check the effect of the stress gradients that way.
Yeah, no, that's an excellent point. We've been trying to understand how that's something we should have thought of, because we've been trying to understand the connection between this cavitation process and fracture. And we've been really trying to sort of understand, because as these cavitation things are growing, these droplets are growing, they're breaking the network. And if you make them big enough, what we know from other people's experiments by injecting needles and inflating, eventually it turns into a crack. So we should have thought of your idea. But yeah, no, that's great. We'll try that. Yeah, there'll be a huge strain gradient there. And
yeah, it's easy to do. Put a crack, just pull it a little bit.
Exactly. Yeah, that's super. I really like that idea a lot. Okay. Anything else? Usually when people stay, it's either because they weren't listening in the first place or they want to ask a question.
Okay.
Our experience is that, 90 minutes is roughly where people start to have sparks going off their heads. When we do this in real life, we can push to 120.
Okay.
But somehow Zoom makes the whole thing.
No worries. As I'm talking, I'm not subject to that constraint. I'm just fired up all the way. But anyway, it's been
one last question.
I have the last one.
Yeah, were we seeing just a small sample of the domain, or indeed, because I saw no edge effects near the sides of your images, and so were we seeing all of it, or just a small subsection of it?
That is an excellent point. And don't worry, Matilda, I heard that you had a question. So so Effie, so usually, we make samples that are several millimeters thick, and
okay.
All of the data that I've shown you are sort of like right in the middle, right? So we're trying to avoid the boundary effects. There are strong boundary effects. And the main boundary effect is this whole elastic ripening thing that I told you about. So if I have droplets in a stiffness gradient next to each other, the material always wants to go to the place where the stiffness is the smallest. So what happens if my material has a free surface, all those droplets near the surface dissolve and we get a puddle on the surface. And sort of over long time scales, eventually all of the droplets will come out and just make one big puddle on the surface. So if we could think of some way of sealing it off so that doesn't happen, if we have really good bonding against it on all sides, I think these things would be truly stable. But as long as they can escape to the boundaries, they will.
Thanks.
Matilda.
Yes, I had a last question. All what you showed was on a specific system with two oil, silicone oil and perfluorinated oil. Do you think that this is very generic and you could find other mixture with where we could observe the same kind of phenomena?
Yes. So we've even done it with pastis. Yeah. So. Yeah. So we tried early on to establish that it really didn't depend too much on the chemical details of either the network or the droplet. And so we made a bunch of different hydrogels. Some were chemically cross-linked, some were physically cross-linked. And we basically used pastis the solvent and then dropped it in water, and we got, you know, these nice little pretty uniform droplets in that system as well. So I think it's, for that reason, I think it's pretty generic.
Okay. Thank you.
Bye-bye.
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 Thanks, guys.
Cheers. It was lots of fun. Thanks for staying till the end.
For us too. Cheers.
Bye.
Bye-bye.